Welcome, everybody. Today it is my enormous pleasure to be speaking to Dr. Ted Steele. And Ted and I spoke last week, although I don't know if this interview is being released yet, on another topic, a topic I suppose more central to your expertise, Ted, as a molecular immunologist, which I'll probably have you explain to my audience in a sec. And what I mentioned to you during that interview is that I had read some of your papers on COVID during the COVID episode. And from there, I discovered your other major interest, which is panspermia. And honestly, it sounds like a bad porno movie. So anyway, so I'll have you explain what panspermia is in a second. But as a broad intro to this topic, one thing I have to tell you is whenever I mention evolution in any of my posts, I get angry comments by people who tell me that evolution is bunk, and it either goes down one of two roads, right? It's either God made the world, that's all you need to know, and evolution is bunk. Or don't you know that Darwin was a horrible racist eugenicist, and so we shouldn't listen to anything that he says? I'm like, I know Darwin was a horrible racist eugenicist, and several of his cousins were way worse than him. But you've got to understand, evolution is not Darwin. There's more to it than that. So my first question is, do you reckon I'm going to lose even more angry people after we discuss what we're going to talk about today?
Well, I would like this to be cool-headed, and in someone like me, I'm not anti-religion at all. So I'm going to just make that first up. It is just as, for my position now as a scientist evaluating the whole panorama of evidence, I've come to the conclusion, and at the end of our interview we could probably sum this up, that it is just as a legitimate understanding or an explanation for the origin of life as the scientist tries to creep towards a scientific explanation. And I use the word creep because it's incremental, but I am one scientist who's covered all that evidence. I'm an objective observer. I try and be unemotional about it, and I've hit the wall. By that I mean is I've discovered that I've got to the point where I can ask precise questions on this scientifically, and I'll explain this at the end, I've reached the conclusion a scientist can never, ever provide the answer to this, ever. Doesn't matter what they say. They can smear and use all these derogatory terms, from atheistic position or anti-religious position based on a whole lot of other reasons. But if you're an objective observer as a scientist, which I like to think I am, you reach the wall and you cannot delegitimize religion. But the trouble is both sides do the smearing. People on the religious side do the smearing, and those on the pure dogmatic scientific side do the smearing. So I'm in the middle. Let me just make that clear.
That is so true. That is so true. And wouldn't it be nice to get everyone in the same room and actually have a measured conversation about this?
Yes.
So what was it about studying molecular immunology that led you to an interest in evolution?
Okay. I've been active scientist, publishing scientist for 55 years. I began life as a mucosal immunologist on my PhD with Professor Derek Rowley at the Adelaide University Medical School. And I worked on mucosal immunity, all the fundamental mechanisms for how the body protects against anything coming in by the nose, the mouth, the eyes, through the mucosal membranes, the mucosal surfaces. And when I wrapped up my PhD, I honestly thought I'd left the field, and I honestly thought all the fundamental principles had now been laid down. Now, in the last 50 years, since my PhD in 1975, that field has matured and added to these foundations, but hasn't changed the principles. Okay, so that's why I gave you the interview last week on the flu shots. Now, major principle is that the two systems, the mucosal immune system and the blood immune system, are completely different, They really are. And you can never, ever activate the mucosal immune system by shooting up the arm, the antigen into the arm, okay? That is a rock-solid empirical fact. So that's why I gained an interest in all this with COVID. Now, when I put that to bed, and all of that matured through the textbooks, the biomedical textbooks, by the way, wasn't just. It was a fundamental, it's the gut-associated lymphoid system, the mucosal-associated lymph, are all there in the textbooks. It's well established, but it's ignored. Now, that is strange. So, but let me move on. I'm a cellular immunologist. I started off as a cellular immunologist looking at autoimmune mechanisms. And then I gained an interest in the Lamarckian inheritance mechanisms, because nothing seemed to add up from my position. And so I was a cellular immunologist and became a molecular immunologist, and then secured over the next 40 years the molecular and cellular evidence for what we now call reverse transcriptase Lamarckian gene feedback loops. It's a real tongue twister, but I'm trying to give you all of the steps in that word. But it was a baptism under fire for me, because I finished my PhD here in Australia. Then I spent almost a year and a half at the Australian National University in John Curtin School. Then I shifted to Canada and did a postdoc. And with Dr. Alistair Cunningham, I was at the Ontario Cancer Institute. And during that period, many things dropped into place for me as a 27 and 28-year-old. And I realised that the Darwinian theory of evolution, through being inspired by reading the books of Arthur Koestler, could not address the fundamental, mechanisms that we need to have for adaptation on Earth. So I severed my connection with. I still live a formal scientist, and I managed to survive for another 30 years in the system, but I formally severed my connection with conventional science on Darwinian evolution. And at age 28, I drafted a book called Somatic Selection and Adaptive Evolution, and I published it. It became a scientific bestseller. It destroyed many relationships with all my scientific colleagues. I could not hold down jobs. But I did enter the circle, the rarefied circle of Karl Popper, Peter Medawar, the great scientist Howard Temin, who discovered reverse transcription, and they all became my supporters. Well, no, one of them was a fierce enemy, although secretly he was an admirer and a supporter, and that was Sir Peter Medawar. He just loved it, but he was a man who'd had a recent number of strokes, and he was looked after by his total support team, so I couldn't have a proper scientific discussion with the man. I was dealing with all these isolates and lieutenants who were absolutely shocked by the data that Reg Kuzinski and I published on Lamarckian inheritance in mice. And that set me on a train of a turbulent career in molecular and cellular immunology, where I had to fight for the very essence of those mechanisms, the reverse transcriptase mechanisms. Now that took me through many battles, many attempts to plagiarise. That was the interesting thing. The people that were trying to bury me were also plagiarising me, so I had to fight them as well. And by the time the late 90s came around, of course, everything came to a head. And I was carrying enormous emotional and controversial baggage at the University of Wollongong. And because I'm an objective observer, I used to call a spade, and I did that in everything I did. And of course, that came to a head with the soft marking issue in 2020 here in Australia, and it was rampant at the University of Wollongong, but on a scale that I could see clearly. It's now rampant in every university, by the way. Soft marking and the corruption of standards is a major problem. Anyway, I was the fall guy. I was the canary in the mine shaft, and after I answered a few questions to a reporter, I'm giving you a very brief summary. I was promptly sacked by the University of Wollongong and the Vice-Chancellor, and that set in train 18 months battle in the federal court to get some justice. I eventually secured that, but it did enormous damage to me. You've got to understand, I could never get a job again in the conventional system. And
so essentially what saying, Ted, is that kerfuffle over the soft marking at the University of Wollongong, that was really an excuse to get rid of you.
Oh, yes, very clearly, because we just published Lamarck's signature, a big book, and it was and they were horrified at the Dr Ted School of Medical Research that Professor Bland and I, who was a senior professor and a fellow of the Australian Academy of Science, were co-authors on the book with Robert Lidley, and it was drawing enormous. So people, I'm going to name some people now because they have to, and they know that through all my other controversial things. But it was basically led by Professor. This whole campaign against me, and the University of Wollongong was softened up, and Gerard Sutton, the Vice Chancellor, just had to get rid of me. You see, there was at that time the international market was, what was it? It was something like $5 billion a year in export market for overseas students, and these are this is so Australian universities basically funding their enterprise through taking overseas full fee paying students. Everything intersected. I haven't finished, but the point was, for the last 25 years, I've still published, and I'm publishing now at a 3-time, 4-time higher rate than I was when I was in the university system, and I'm publishing in peer-reviewed journals. And by the time of 2015, I don't want to go through all of the controversies and battles I had to fight, but they've been numerous. I've won them all, and that's part of the problem, that I won them all. And when you win your battles against vehement enemies that want to expunge you from science, you accumulate more enemies for some reason. Anyway, so in 2015, I joined with Chandra Wickramasinghe, who with Fred Hoyle had gathered all the evidence on and marshalled it and critical demonstrations of panspermia. So panspermia is the seeding of life.
Tell us what panspermia is, okay?
Sorry, you can see I had to do that. It was 55 years, so not as if I'm a spring chicken anymore.
Yeah, and I think I'm glad that you outlined this process, because I draw two things from it, and I hope my audience does too. Number one thing, Ted is not a crazy person. He actually has a very solid background and from reputable institutions, and is well published. Second thing, scientists do not float on clouds. They are insanely jealous. They are competitive. They glom onto dominant ideas and get really bent out of shape when someone brings along evidence that actually challenges their paradigm. And I don't think my audience is going to be shocked by that second thing, but it's nice to underline it. All right, so now we get down to it. Panspermia, crazy word. Give us a definition of it.
Yeah. I was aware of it. I became aware of it in the early 90s, but joined with one of Hoyle Wickramasinghe. Hoyle's dead now, but Professor Chandra Wickramasinghe in 2015. So I've been in the field for 10 years now. Whilst I'm a molecular biologist and I'm not an astronomer, I've learned a lot of astronomy, basic astronomy. I've learned a lot of conceptual analysis. And it's very clear, reviewing all the evidence marshalled by Fred Hoyle and Chandra Wickramasinghe over 50 years, that the universe is in fact teeming with life, and life from the universe in various manifestations and forms has seeded life to the Earth. As soon as it cooled down, as soon as it was ready to accept life after the Hadean period at 4.2 billion years ago, as soon as it was ready, it was a niche for life to flourish. And life was raining in. The seeds, as panspermic seeds of life of various and multiple different forms of life were coming in. It began to flourish. And so that is panspermia. That's the essence of panspermia.
Yeah, breaking the word down, pan means everywhere, right? Yeah. Like universal.
Yeah. Seeds.
So spermia is part of the seeds.
Like sperm.
Yeah, got it. Absolutely fascinating. So you mentioned that this panspermia hypothesis was first put forward by the really quite legendary astronomer Fred Hoyle, and I believe Chandra. So I'm going to stumble on his name. Apologies. Wickramasinghe.
Yeah. By the way, it was joint effort between Fred and Chandra. Oh, I know the history of that collaboration as well, and the publication history. It was a truly joint effort. They were both creeping towards it, and it was Chandra leading the way as a postdoctoral student.
Yeah. So he was Hoyle's PhD student, or
he was Hoyle's PhD student. Hoyle had written Life Cloud. Or the Black Cloud in the 50s, which was a fictional account of life in the universe. So Fred had already reached that position, but you see, the greatest scientists can also do other things. They can be excellent communicators. They can be writers of incredible perceptive novels, like that novel was. But it was recognised as a novel. He was knighted in the 1950s for his contribution to science. He was an icon of British science. Then Chandra joined him, and Chandra was given various tasks to do with astronomical observations on dust and all that sort of stuff. But the point was, Fred was always sceptical. He's a bit like me, actually. I'm very similar to him in that way. You need the evidence before you can make the leap. You can dream and think about things and speculate, but you've got to need the evidence. And they were both of them creeping, but Chandra was leading the way. We've got to make that clear. In the critical first papers, Chandra was the sole author. Fred was an honourable man. And it was a truly joint collaboration over 50 years. So we've got to use both their names. If it wasn't for one, it wouldn't have matured, and it had to be they had to both. But Chandra is a great scientist in his own right. His output is extraordinary.
He's still publishing to this day.
So it's very, very. So they were creeping towards it in the 1960s, and then by the 70s and the space age emerging, they really firmed up some of their predictions. So I'll leave it there now, because I know you want to ask me something else.
Well, yeah. What I'm curious about is given that both Hoyle and Wickramasinghe, I'm getting better at pronunciation, thank God. Wickramasinghe. That's how I remember it. Wickramasinghe. Okay. Given that they were both astronomers, I suppose my question is, were they coming at this from the perspective of we think there are holes in evolutionary theory, or?
No. I don't think they were, actually. No, I don't think they were initially. But it dawned on them that was their huge obstacle they had to deal with. But I can't be certain. I can't be certain of that because I wasn't around. But just reading between the lines and the way their papers evolved and books, I can't be certain of that. But let me say this: they were both hard-nosed, sceptical astronomers and scientists, but they founded the new science of astrobiology. There's no question in my mind. They had forerunners. They had the Swedish chemist, savant Arrhenius in 2008, and they had Lord Kelvin in 1894, and prior to that, Louis Pasteur gave the seeds of it. But the real marshalling and then conducting direct tests, that's a critical thing that separates science from speculation and non. You make a very specific prediction, and that's really important. They did that. Then you ask, can I find evidence for that exact prediction? Now, we're talking about exactness now because we're dealing with physics and astronomy. We're not dealing with a lot of the rubbery things that happen in biomedical science, because living systems are co-complex. To try and study them the way a physicist studies innate matter is almost impossible. That's why we have so many conflicting reports of fraud and failure to replicate and all that sort of, because living systems are extraordinarily complex.
Yes.
And if you don't understand that, you can't understand all of the controversies that are raging all the time. The one-size-fits-all controversy, for example, that all human beings will respond to a medicine in the same way. They don't. As you know, I'm sure you know yourself, every human being has its own personalised response to whatever it is. Anyway, look, I'm deviating a bit, but the point was, in the 70s, they started to really do the hard science of prediction, securing evidence, looking at the prediction, does the evidence, and then it was independently confirmed, by the way, years later. I'll get onto that in a minute, but the point is they were hard-nosed, sceptical scientists who wanted to do what scientists do all the time: find a signature for life on Earth shine their telescope into the interstellar dust and the cosmos, can we find that same signature out there? Now, every day, and this comes from the periodic table, all the elements on the periodic table have a particular emission and absorption spectrum over defined wavelengths, right? They all do. Distinctive. So you can tell whether it's hydrogen out there or carbon out there or a molecule, and complex molecules have their own distinctive signature. So it's a standard of astronomy, have the signature of all this basic stuff on Earth, and shine your telescope, whatever it is, a radio telescope, the James Webb telescope, any of them, and ask, can we find that signature out there at a specific location in space? It's a standard way all our knowledge of astronomy has been built. Now, that's exactly what and Chandra did, yes.
Yeah. In other words, when astronomers look for the possibility of habitable planets, based on our understanding of the conditions for life on Earth, they're looking for things like water. Exactly. Yeah. So you're saying, so that's a molecular signature, and you can do the same thing with let's just say more complex life forms. Before we get to that part of it, I want to talk about abiogenesis, because this to me seems really, really central to panspermia. So could I start by having you just define what abiogenesis is and where this fits into the Darwinian evolutionary framework, let's say?
Yes. Abiogenesis, that's how I would pronounce it. Abiogenesis.
Oh, I'm sorry. Yeah, abiogenesis. Okay.
Yeah, and other people may, that's the way I pronounce it. The thing is that if Darwin first posited this in the late 19th century or the mid to late 19th century, that life began in a warm little congenial pond somewhere on Earth, 3.5 billion years ago, and then all the elements and the ingredients in that warm little pond came together spontaneously to assemble. And let's face it, we have to be real, not just a bit, but a living cell. That's the key thing. A living cell is one of the most complex, as you know yourself, one of the most complex microscopic structures. And of course, whole organisms are built on these, But anyway, a living cell is extraordinarily complex. And Fred and Chandra, both of them did this, by the way. We've got to understand that all of these calculations that I'm now going to give you were both Hoyle and Wickramasinghe, probably led by one time and led by that, but they both did it together and confirmed, because they're both mathematicians. You've got to understand, they're mathematicians, astronomers, and they're founders of astrobiology. It's extraordinary. They have what they have done. They are in the pantheon of great intellect.
Yeah, yes. The number of fields that get integrated into this, and I want you to talk about the calculation because it's fascinating. But the thing that I want to preface this with is just saying, okay, when critics of evolution do their criticism of evolution, this is one of the key barriers to overcome, right? It was the idea that there was a big bang, and then there was primordial soup or whatever, and there was a bunch of elements that were just doing element things, and then purely by chance those elements combined and then formed amino acids and enzymes and carbohydrates, and then assembled them into cells.
I know, but I just add this. It springs up all over the universe to explain all these sightings they're seeing now in the exoplanets. The theory has been matured under a Big Bang theory. It's been matured to the fact that this abiogenesis event not just occurred on Earth, but occurs conducively on all planets. Okay, so that they don't want to talk about cross-infection or spreading of life between one place and the other in the universe, which we do, but. So abiogenesis, let me give you the probability now. Based on the fact that there are 20 amino acids, based on the fact that there's an average, they pick the conservative estimates have several hundred, average protein of several hundred amino acids. I'm technical now, but I'm sure some of your audience will understand. But then, if you line up a lot of proteins that have common functions across the spheres of life, you find, for example, hemoglobin or hemocyanin, or a whole range of different protein, myoglobin. There are constant invariant amino acids spread throughout the through a protein, which gives us its three-dimensional structure, so it carries out its catalytic function. Now, they just made a couple of, just back of the envelope, conservative estimates. Let's just assume that there are 10, for each protein, there are 10 conserved sites that have to be conserved. You can let all the others vary, but if you don't conserve those, you won't have the structure of the protein. That's reasonable.
And if the structure doesn't have its protein, it doesn't work. It doesn't do whatever it's supposed to do.
Yeah, that's right. Then we have 20 amino acids. Okay. Now, they're mathematicians and they work out probabilities. They came, okay, that gives you 10 to the 20, right? So then they asked the question, how many basic genes would you need for the most elemental cell that we know of, and from all our knowledge now in molecular biology, to produce the first elemental cell? If that turns out to be, that's called the Kunin number. He's the Russian scientist at NIH in the United States that actually helped refine this clearly, and it was a very important number. There are basically 256 basic proteins. If you don't have those, you don't have a mature cell, okay? Okay, so you can see we're being very conservative. So we have 10 to the 20 times 10 to the 20, 10 to the 26. We have 10 to the 5,000. So the probability of getting
5,000 zeros after it. Okay.
Raised to the power 5,000 times. And the number, and of course, this is why critics of science, deride it. And fair enough. So do I. You got to understand, I'm in the middle.
Would you buy a ticket in a lottery that had a 10 to the power of, even 26 chance of winning?
Well, let's put it in perspective, right? Okay? The Big Bang universe out to 13.5 billion light years, right? I'm going to talk about that later because it's no longer that. It's collapsed. That whole theory's collapsed. But that just in the Big Bang observable universe, there are 10 to the 84 protons. Now, what's a proton? Proton is the nucleus of a hydrogen atom. The most dominant atoms in the universe and the building blocks of all other elements, if you go to the periodic table, are hydrogen atoms. And so that's a very important number. So 10 to the 84 in the total Big Bang universe. And we're talking about a number of 10 to the 5,000. 10 to the 5,000 not only outstrips the molecular resources, but it therefore has to outstrip all the statistical resources of the Big Bang universe. So it's impossible. It's impossible. It couldn't have happened, right? It would not have happened. That's the point I'm trying to make.
Yeah. And if Darwin proposed this as the origin of life on Earth, primordial soup, elements, abracadabra life, it's not possible. It's not possible for it to happen.
Darwin didn't have any of the sort of knowledge we have today.
No, of course. I mean, they hadn't even discovered genes at that point. Yeah.
The people, the Big Bang people, origin of life people, all cling to this assumption of Darwin. It's an extraordinarily porous and sloppy, ill-thought-through assumption when you come to the sort of probability number, which is based on hard facts, by the way. Those 256 essential genes, that was arrived at not easily, and Eugene Kunin, and his collaborators arrived at that number. And, of course, that actually helped Fred and Chandra modify their number. They originally had 10 to the 40,000. Then they realised, no, let's be more conservative. Let's go to the essential number of genes required, protein-coding genes, to get the first replicating free-living cell, because that's what you need in any origin of life theory on Earth. And it's 10 to 5,000. Now, look, that explains why all the attempts to cook things in the lab and get life emerging from, the elements have failed. The ones that have been successful, and by the way, this is pretty embarrassing, Nobel laureate picked up his laureate in one area, they went into the origin of life area and discovered, and he was doing very, very precise experiments in the lab with cells and that to get the first self-replicating RNA molecule. And look, he was a serious scientist, and then he had to do the serious thing and retract all his data, because in a year or two, it was clear he had to retract it. They could never reproduce what they'd done in their cells. So he did the right thing and retracted it. Now, that is the most serious attempt to do it. and it had to be retracted. Every other attempt,
serious attempt. In other words,
negative data. This is totally. It's the 10 to the 5,000 says it can never be achieved. You see.
Yeah. So again, just for my audience's clarity's sake, what he attempted to do is to take the elements, the basic building blocks of life, and then add heat and I don't know whatever else, and see. And he couldn't. Newsflash.
No, the early ones, the Uray experiment, those experiments in the early 1950s. I've forgotten the names. I've just forgot the names of the key authors. But where they put up a chemical soup in a flask and then hit it with flashes of light and high energy radiation, and to try and conduce the conditions that might have been on the early Earth to produce just some amino acids. They did in fact actually get some, but they got no life.
Yes, they got a building block of life, but not life itself.
The fact that they, by forcing it, they got some. Said, oh well, we're on the right track. You see what I get? Look, all of that work has fallen over, in my view, as an objective observer. It's totally worthless research. In fact, I have to say now, after 50 years, that billions of dollars of physics research has been spent on this exercise. It's totally wasted. So you've got to, at some point, you've got to say, let's be reasonable. Let's just embrace the 10 to the 5,000 and realise that is a barrier now to scientific knowledge. You see, I'm going to talk about barriers several times now because that's a big one. The 10 to the 5 is a big barrier. We will never, even if you want to trim it down from 256 and get it down to, say, 100 essential proteins, you're still going to get this big number. Do you see what I'm... So, look,
it's still basically...
Abiogenesis as the origin of life on Earth or anywhere propping up spontaneously throughout the universe is simply off the table. As a scientist, I accept that. It's an unprofitable scientific exercise to pursue that now. We have enough evidence. It's totally wrong. Okay? I can be really definite about that. Yes.
And so if the predominant hypothesis, this abiogenesis hypothesis, basically it just hasn't panned out, then it would make sense to look elsewhere.
Alternative hypotheses, yes.
Yeah. Put forward alternative hypotheses, and then you generate predictions based on those hypotheses. You go out and either run experiments or you look for evidence and see if your predictions are confirmed or not.
And that is what Fred Hoyle and Chandler were saying to you.
Yeah. Okay. That brings.
Go on. Sorry, I just have to say, as a scientist who understands that scientific process, on the battle for Lamarckian inheritance, which we've won, by the way, this is why I conceded. It's an incredibly difficult fight. So all I want to say is that they are behaving scientifically, making predictions, and we'll show some of that evidence shortly, I think. And so that's the way scientists behave. If you're getting dead end results across the board, in the end, when you figure it out, well, we can never actually achieve this, you've got to drop the research program and do something else that's more likely to achieve a more positive result. That's the only advice I would give the mainstream. But I don't think they're listening. But I think they are listening, by the way. They're listening all right. They just don't want this voice to be heard.
Yeah. It's the old science advances one funeral at a time, right? I mean, You've got people who have careers and probably Nobel Prizes and university grant funding and God knows what else. Their reputation.
Industry based on it. It's an interlocking industry. Make no mistake.
Yeah. It's very similar. It's very similar to what we were discussing in the other interview on flu vaccines. The fact that, all right, as a mucosal immunologist, you know damn well, and so does St. Anthony Fauci, that you cannot have a successful vaccine delivered via a shot in the arm for a respiratory condition.
So it's highly offensive in 2021 when I realised what that vaccine, what it was. I just thought, they're not going to work anyway. They're harmless. But then the mRNA changed my mind. But that's
That's another story. Okay. Abiogenesis is dead in the water. It's just mathematically impossible.
Yes.
What's the alternative? Yeah. What's the evidence? What's the evidence for the alternative?
Let me go through the evidence for the alternative. And I've got 2 slides I want to show you. And by having them up and then me talking to the slide, I'm sure that those who are listening and watching this that are really interested will understand what I'm saying. Okay?
Perfect.
I've had a bit of practice now describing this slide, but I actually contributed to one of the later phases of this slide by working on the probabilities for the independent confirmation of it, which
which one do you want me to put up?
I'm going to use words like independent confirmation a lot, because that is the gold standard of a scientific fact. Do you understand? If other people, totally independent of you, from a different angle, can get the same result and the same conclusion, you've got to accept it. Many scientists don't do that. That's when you've got to change your mind, by the way. And you've got to change your mind.
Yeah, this is replication, and anyone who's
and that's the interstellar dust slide. You know, the one with the.
Sorry, was that the Elan Vital? Was that one?
No, that's going to be the last slide.
Okay, infrared flux.
That's it. That's one. Yeah, you get that one up. Yeah.
Perfect. All right.
Okay. If you don't mind, Robin, I'm going to spend a few minutes describing the slide in detail so everyone understands the evidence and the implications. Okay. This slide, this data was secured between about 1980 and about 1983, and it was secured in 2 phases. Now, first of all, there was a first phase here on Earth, working out what the signature is. Then the next phase was getting out into the cosmos and shining a telescope. Okay, that step. Is this signature in the cosmos? Right. Now, Fred and Chandra in the late 1970s made the following prediction. They said, what if we establish on Earth the absorption extinction signature, which that's what relative flux is here. That is, when you shine light over a certain wavelength range, like in this case here, this is the mid-infrared range of about 2.9 lambda microns. A wavelength of 2.9 micrometers to up about 3.9. That mid-infrared range of wavelengths, if you shine that through freeze-dried bacteria, right, that are not dead, they're dormant, they're alive on Earth, because that was the prediction. Maybe space is filled up with freeze-dried bacteria or something like it, because of the temperatures of deep space are, minus 273 Kelvin, they're absolute zero. So in space, everything would be frozen in deep space. Except in aggregated niches like planets or central portions of large interstellar dust clouds where it heats up a bit, okay, through the activity of life and just the molecular collisions that are going on. Anyway, let's get back to this figure. So on the x-axis, we have the extinction or relative flux, right, of shining this reddish light through in a flask, in a vacuum flask of freeze-dried bacteria that are alive. Okay, you've got to understand that. And this was done by Sherwin Al-Mufti. If you look at the little box at the top there, he was a PhD student at Cardiff University in the Institute of Physics there that Chandra was heading up at that time. This is before they were banned as academics. And Fred and Chandra got Sherwin Al-Mufti, he's now in his mid to late 70s, he's still alive, got him to establish, see the solid line? If you look at that solid line in the figure, going from about 2.9 on the y-axis up to about 3. The solid line, it's a solid line. That is the line of the spectrum of the absorption, whether it absorbs or not. That is, if it absorbs the red light, less light gets through to the detector. If it doesn't absorb light, then more light gets through. Did you understand the principle? So that's
So you're shining a particular wavelength of light, and some of that light is going to be reflected back from the organism, I presume.
Absorbs, it absorbs, so it doesn't reach the detector. You see, when you shine light from this,
Okay,
right now. That's a stand. This goes on all the time in the lab, in medical labs, physics labs. You know, I did it in my PhD. You use OD, optical density or extinction, to measure concentration of all sorts of things over defined wavelengths, proteins, bacteria, the lot. The point is, Sherwin Al-Makdi established this signature over defined wavelength range. And if you're an astronomer, that means a lot, right? 2.9 to 3.9 micrometers. So that means anyone else can go away and do this, right? But look, we're not just talking about one point, one wavelength. Look at the range. It goes. There's numerous points built that solid line. Forget about the experimental, sample
so
points.
Each of these little dots here is
that is the experimental. That's the test in the cosmos.
Yes.
The solid line is what was established in the lab. Okay.
I see.
Yes, you see, you get it now. And that's numerous points. So it would be 2.91, 2.93, and numerous points established in the lab. You get a printout over that wavelength.
So let me get. What I want to do, I'm just going to pause you there and see if I can explain what you've said so far from a lay perspective, and then you can correct me. So, okay, you're shining wavelengths of light, and each wavelength is there's just micrometers of difference between them, but these organisms absorb differing amounts of each wavelength of light.
That's right. Yeah, and it's a reproducible signature for freeze-dried bacteria, but you can see it similarly in many other freeze-dried cells. But this is E. coli, the one we all, the whole workhorse of molecular biology. So this is a standard organism, and most people would accept that. If you didn't have all those scattered dots there, just that solid line, that's reasonable. That looks like that. That's what you would call the spectrum, the absorption spectrum over that wavelength, right? But then this is the prediction. This is the famous scientific prediction that Fred and Chandra did. They said, we predict, this is the announcement they made, we predict that if we shine the telescope over that and pick up an emission source at the centre of the Milky Way galaxy, which is 23,000 light years away, and focus on that emission source, GCIR7, over that wavelength range. A physicist knows an astronomer knows what I'm talking about, even though I'm not astronomer, I'm communicating in their language. Then we should see over that wavelength the same signature. So let's go out there now, and Dale Wickramasinghe, you see in the author list at the bottom under the figure in the legend, Dale Wickramasinghe and Dave Allen working at the Australian National University, and then using the Anglo-Australian telescope at Parkes in New South Wales, they got telescope time. I don't know how they did it, but it was a bit of subterfuge, I think, involved.
It might involve liquor. That's just my guess.
Anyway, they got it, and they had a lot of time on it. And so each little data point there, you see, it's got a little red dot with it, and where it's variable at a different wavelength, it's got an error bar right to it. But look at the dots, how they cling to the predicted line of the signature on Earth. Look. That is the extraordinary result. That is what's called an exact match and fulfillment of prediction. The probability of getting those dots from that source, 23,000 light years away, and the light would have, over 23,000 years, would have been coming through many thousands of different massive interstellar dust clouds, to get to Earth. And these are the fine grains. Now look, the probability of getting that by chance is something like 10 to the 13, 10 to the 14. It's just, again, another impossible number.
Impossible with too many zeros. So again,
Go on.
My layperson summary is, so you find this, essentially it's a molecular, no, it's not a molecular signature. Would you call it a light signature?
Yeah, it's a molecular signature. It's the molecular signature.
And then you point a telescope out into space. Yeah, so you point a telescope out into space, and what your I don't understand the mechanics of this well enough. That telescope is projecting the same infrared light. No,
it's picking up the radiation from this source. 23,000 is highly focused on it, and it's asking how much of that signal over this wavelength range is getting through to our detectors here on Earth.
Yeah.
Okay. You see what I'm getting at?
Yeah.
It's an absorption signature, the extinction signature of the sort I was describing before.
Oh, so, now it's clicking. Okay.
This is a rigorous test. A lot of people don't. I'm glad we're doing this at some things. Look, physicists don't want to know about this, by the way.
Yeah.
Hard-boiled scientists don't want to know this data. You try and explain it to them, like I'm trying to do to you now, and I've already had Zoom meetings with them, and I couldn't believe I was dealing with physicists.
Infrared light of these wavelengths is out there in space. It's produced by, in general terms, space phenomenon, and something is absorbing those frequencies of light in the exact same pattern as freeze-dried
on Earth. Yes, on Earth. Now, I've just explained to you how astronomers build up their knowledge of the universe, by this chemical knowledge. This is the same principle. They establish the signature over a wavelength range or whatever on Earth. Yep. It's the same principle. It goes on all the time.
So this is how you would detect the composition of the atmosphere of Saturn or whatever.
Yes, exactly. Work out that there's carbon dioxide. Well, Saturn has a range of gases, but an exoplanet. This is how you would be able to get some idea of whether that exoplanet has water, would be congenial to life, and also water has its own signature. All of them have their own signature. So the point is, there's nothing unusual of what Fred Hoyle and Chandra Wickramasinghe did here, and with their student. This is a Nobel Prize winning experiment, by the way. If we were living in a normal world, this would be a Nobel Prize winning experiment.
Yeah. Taking a tool. They're taking a tool that astronomers use all the time. To detect the presence of things that we have here on Earth, elements and so forth, out there in space.
Yeah, and see how complex it is, Robin? See how complex that signature is? We're not talking about one point. We're talking about numerous points, all clicking to a line. The probability of getting by chance is an impossible number. This is an exact match. Now, 9 years later, after Dave Allen and Dale Wickramasinghe, Chandra's brother, a mathematician and also a part-time astronomer, and an astronomer. But that family is an extraordinary family, by the way. The whole family. There's a whole family of geniuses in the Wickramasinghe family. It's another issue. It's an issue to do with genetics. But the point is that 9 years later, a group in Japan sampled the same wavelength range at the same site, GCIR7. IRS7, with another telescope in a different way, and they got exactly the same match to the predicted line.
There you go. There's your
objective fact I'm giving you here now. It's nothing to do with made up or pure speculation. This is an objective scientific fact. And I have to accept it. In fact, I saw this sort of data about 30 years ago and nodded my head in quiet agreement. I was reading Fred Hoyle's book, Our Place in the Cosmos. I recommend the book, Our Place in the Cosmos by Fred Hoyle and Chandra Wickramasinghe. That plot is in that book, but it's a wonderful book. And if you've done any basic, bachelor's knowledge in microbiology, you'll understand it. The point is that is an iconic experiment. Now, let's just not leave it at that. But by the reaction, I just have to give you the reaction. The reaction led by NASA, of all people, has been smear and discredit for 50 years.
That's so interesting, because you know that NASA stands for not a space agency.
Well,
that's the going joke.
Summing up at NASA, they've got very good engineers, very good physicists, technical, but they do not have any biological nous in their body at all. But I think it's deliberate. I think the smearing is deliberate because of the deep implications for religion, the National Security Council in the United States. So it's been deliberate, systematic smear of anything to do with this. That's why we see all of these major websites smearing this whole idea. It's not just by chance that it happened. Anyway,
I was doing some reading to prepare for today's interview. Yeah, the descriptions of papers that you've written and that Hoyle and Wickramasinghe wrote, it's I mean, there's such an insulting tone, and it's a very similar thing to what we encountered during the COVID episode, which is who's wrong? This idea is stupid. Okay,
but
you give me some evidence. Why?
The world is waking up to that. Not everyone in COVID, but this one here is the big one. This is officially sanctioned smearing and suppression right at the top. The Chinese are embarrassed. The Chinese, I think, probably know it's true, but they're embarrassed. Indians have confirmed some of things, space predictions for Chandra, and they're probably embarrassed. You see, there's huge international strategic politics involved in this whole issue. We're not just talking about garden variety. You know, people like me are probably more like Mr. Magoo, walking through there and thinking, why are they all so angry? But let me can we go to the next slide on the meteorites? Because the
meteorite fossils, this is mind-blowing. Yeah.
This is, and the way I'm going to do it, on the screen, I've got photographs of 4 of the, let me explain how this is done.
Yep.
These are 4 meteorites. The first one is the Orgueil that fell in France in 1864.
Yep.
The second one is the Migi that fell in Ukraine in 1889. The third one is the Murchison one, which many in Australia fell outside the town of Murchison in 1969. And the fourth one is the most recent one because of Chandra's involvement with the institutes in Sri Lanka. The farmers, witnessed this fall, the Polonnaru meteorite, just in Sri Lanka, in paddy fields. Anyway, let me go through them and let me explain how the work is done. The first point, I'm showing you evidence of what they see inside the meteorite when they crack it open. Okay.
Yeah.
What they see inside.
That's important. We're not talking about things clinging to the outside of the meteorite that might have been picked up on its way through or something.
But we know it can't be contaminants. I'll explain that too. So that's the first thing. We're looking inside the meteorite. In these 4 meteorites, you see all the same things, which is quite extraordinary given the fact they're all independently fell at different times, but they're all carbonaceous meteorites. That's the first key point. They're not rock-hard asteroids. The word asteroid is used a lot to deviate attention from that's another political decision, to shift attention away for the fact that all of these are carbonaceous meteorites. Now, the word itself implies something. It implies they're carbon rich, which they are compared with the stones and the steel asteroids. So that's why they never want to talk about cometary carbonaceous meteorites because of that. Isn't that extraordinary? Yes. Politics happens right.
The important being, life on Earth is carbon based, right? Like that's
exactly. So that's the first thing. But they're loosely held too. We might come to that later. But they're loosely held carbonaceous meteorites. So and that's because they've got their snapshots of fragments from comets, real comets like Halley's or the others, that and the Earth passed through the meteorite fragment streams of these comets, and we've picked up some of these meteorites, and they're carbonaceous meteorite. Now, in them, you can see extraordinary detailed scanning electron micrograph evidence of cells, bacterial cells, and not just bacterial cells, eukaryotic cells. So in this fragment, let's just take the one at the top, the Orgeal, 1864. On the right photo, that is a diatom. A diatom is one of the silica shelled, the glass shelled phytoplankton that inhabit our oceans on Earth.
Yes.
And the others are, very complex bacterial species, right? But then you go to, but these things have been seen in all of them, including the most recent one. The point is I've got to go through some other stringent things here, other tests, because these are the questions I was asking when I was confronted with this. Have other objective observers, independently of the original claim, gone into the same meteorite and seen the same things? Mm-hmm. The answer is yes. I've got to say that now, yes. Secondly, how can they tell that they're not contaminants? Well, Richard Hoover, one of the leaders in the field in this, he's worked on the Polonararu a meteorite, and Murchison in particular. They with a scanning electron micrograph, it's not just like any ordinary microscope. It's interactive. You can get information on the chemical composition of your target area, and as you scan over an area, you've got a figure there. You see those highly detailed cellular structure. As you scan over the cellular structure and then go into the surrounding matrix, the medium which is supporting this fossil, that's what it is. It's a fossil. You ask yourself, is the same chemistry the same, basic mineralized chemistry? And the answer is yes. Now, that is very important because all fossils are mineralized from the surrounding material, you see. Uh-huh. So it couldn't be a contaminant. See what I mean? We've got to be logical about it. It can't be a contaminant.
Got it. It was fossilized within this rock that it's hurtling through space on.
It's inside the rock. This preserved fossil of life is inside the meteorite. Now, the third thing, really important, all of these carbonaceous meteorites, and many others, by the way, but the carbonaceous meteorites are really important because they come from fragments of comets. These are, and comets in many ways are aggregation of smaller carbonaceous structures. I won't get into that, but the point is they're dated. This is important. How old are these? How old would the scientists agree these meteorites are? See, if they're 3 billion years old, that means it's fallen to Earth. That means it could have been, a contaminant on Earth, or that it appeared miraculously on Earth. The trouble is, all of them have an age greater than 4.5 billion years before the Earth was formed. Now, this is very important conclusion. It is so important, it upsets people. It says that mature cell-based life, to me, I'm an objective observer, I'd like to think I am anyway, mature cell-based life of bacteria and higher cells, the eukaryotic cells, the building blocks of our cells, are in existence as captured fossils in the solar system, before the solar system formed. That's what that says. That means...
How's the dating?
That is demarcation. For me, there's no other interpretation, you see?
How is that dating done? Like, how do we know that these
they have various ways of doing it. I'm not a physicist, but I do know that highly rigorous testing has been on the Murchison one, because that was one of the most recent, right, in Australia. So whether it takes away the fact that they're not 100 years old, but we're a recent one. To be honest, you're going to have to go to the physicists, but I've read their papers. I don't fully understand their papers, but they all agree, these are in Proceedings of the National Academy of Science, but they all agree that this dates it. In the case of the Murchison, they reckon that some of the gases or other things that they're detecting in the little parts of the meteorite date it to not 4.7 billion light years, but to 7 billion light years. So sorry, 7 billion years, not light years. That's
7 billion years.
7 billion years, well before there was even a gleam in the eye of this aggregating solar system that was going to be eventually our solar system and the Earth. So it's much. So the point is, they used techniques, chemical techniques, radiation techniques to do the dating. You know, it's not like carbon dating now on Earth. They have to use other specialised techniques. But the important thing is they agree on the dates, and it's just agreed that they are. It would be very surprising if they weren't now. If people started to come up and claim there would be lots of scientists who have put their career on this line of getting the date right, which would argue with them. But there's been no dispute about that. You've got to understand. Now, but the reaction to it, that's where, you know, what's happened with all of the reporting on the carbonaceous meteorites with respect to microfossils within them has been highly emotive, smearing. I am staggered by it. I actually confronted it in a Zoom meeting two years ago with a whole bunch of physicists. And it even penetrated down to this group who were very interested in the origin of life in the universe. And one or two of them had to admit this, look, everyone agrees on this data that Ted's bringing to our attention. Why are we disputing it? And that silenced all the others who were trying to question, you know, my credibility. So you see what we're up against? And you can see why I talk about this, because I've met this with Mark and Darwin. I've had the same problem, and we saw it with COVID, that the cognitive shift required to get your head around the evidence and all the other things that you thought were true about the world, you have to now abandon. You see what that's implying?
Absolutely. And I mean, we see parallels of that just in all sorts of other controversial situations. But the implications of this are so vast, aren't they? I mean, it's not just scientists' careers built on this. It's people's worldview. It's
people's worldview. And really we're going to come to that because I've thought very deeply about it. Anyway, but look, you go ahead. You keep summing it up. You're doing great.
Yeah. No, okay. All right. So what we've got now, again, just going back to how the scientific method is supposed to work is you make some observation, you form a hypothesis, you make a set of predictions based on that, and then you design experiments or make further observations to test those predictions. So what we have now is a hypothesis that life came to Earth from space, from the cosmos. Yep. Okay. Life came to Earth from the cosmos. And so the predictions based on that would be that you could find signatures of life here on Earth out in the cosmos. So that prediction is upheld, and we saw that from the molecular signature chart. The second prediction based on that would be that objects that are hurtling through the cosmos, like comets, would then contain life. So the prediction based on that would be: will you find evidence of life inside meteorites coming from those comets?
Which are cometary fragments. Let's get that clear. The carbonaceous meteorites are cometary fragments of comets. Yes.
Gotcha. Thank you. That's a crucial step to fill in. Yes. And so now that prediction is confirmed.
Yes.
So
two critical demarcation predictions have been confirmed. Okay.
What? Yeah. I mean, we've touched on the implications of that for people's entire worldview and scientists' careers and Nobel Prizes and God knows what else. What are the implications of that then for evolutionary theory as let's call it mainstream evolutionary theory as it is taught in universities and practiced by evolutionary biologists, let's say?
Yeah, very good point, because that brings in my past career and ongoing, by the way. I haven't given up molecular biology. My major research interest is cancer research, by the way. It's not panspermia. It's establishing the mutagenesis signatures in cancer cells. Again, reverse transcription, the same signature I talked about. I built everything else on the somatic hypermutation mechanism, which is now turning out to be true. All the evidence now, hard evidence, supports it. There's just a dead silence out there because, Ted's right again. Look, I'm not rousing myself up. It's just that I'm an old guy now. I've got not much to... I'll talk lucidly about it, but that's the reality. I still live in that total denial zone. Anyway, all these things, Chandra said to me in 2018 after we published a big paper in Progress in Biophysics and Molecular Biology on the origin of the Cambridge explosion. That was our first big paper on this. But he said, look, Ted, all your work on Lamarck, we've got to fuse it with panspermia, because I agree with him. And Reg Wazitsky, my collaborator, he's still alive. Reg and I are still publishing together from the 1970s. We agreed. We said, yes, look, Lamarckian inheritance now, which we can show, occurs in many, many earthly systems. It's been more than confirmed in my mind. It's just that no one willing to stand back and look at all the evidence.
Let me pause there. Let me pause there, because there is an early conflict in the development of evolutionary theory between Darwin and Lamarck. And so when you talk about Lamarckian inheritance, so just flesh out that story in brief. What's the elevator version of that conflict?
I'm going to talk about Lamarck versus Darwin now. Okay.
Yep.
Darwinian theory is ponderously slow. That's the first thing. It's random mutation and natural selection of the survival of the fittest, right? We're all familiar with that. It upsets all of those that can see that, particularly religious, spiritual, can see that life is far more than that. But and how could life evolve? How could the embryo grow through all this stuff? Look, the Lamarckian theory is a little bit older than Darwinian's theory, but Darwin, I've got to make a few things about Darwin. Darwin was also a Lamarckist, you've got to understand. And Darwin's father, Erasmus Darwin, was a rampant Lamarckist. That is, they believed in the inheritance of acquired characters. It was quite respectable in the 19th century to believe that Darwin was right and Lamarck was right. The problem was Darwin himself, his ego got in the way, and he could see that Lamarck was a competitor to his grandfather, and so he had to start the smear against Lamarck, which I'm a little bit, he's gone down in my estimation a bit, Charles Darwin, because I think he did some very important things. Anyway, the whole idea is this, and this was solidified in about 1881, 1883 by August Weismann, the German biologist, who erected what's called the Weismann barrier. And that's where I came in as an immunologist into Lamarck. The Weismann barrier is a barrier around our reproductive organs that prevents any information, acquired genetic information in modern terms, coming from the body cells, to the reproductive organs, entering the germline, registering there, and then being passed on to offspring, okay? So that barrier was erected, so that forbade Lamarckian inheritance of acquired characters, that is, as an organism.
Sorry, there's something I don't understand. When you say
August Weismann.
How is the Weismann barrier actually established?
No, it's porous. It's totally permeable and porous. That's the point.
So it's not really a barrier.
No. It's selective. I'm certain it's selective, but it's porous.
Okay.
And our big paper, if you go to my website, you'll find it's called Lamarck and Panspermia. And we go through, it's a big paper. We go through all the evidence for panspermia in the first part of the paper. Then we have a section called the demarcation zone, where we go through all these other boundaries and probabilities. In the first part of the paper, we talk about Lamarckian inheritance versus Darwin and show conclusively that Lamarckian inheritance has now displaced the Darwinian theory of evolution on Earth. A whole range of different evidence, by the way, does that. I don't have time to go through it all, obviously. But the second half of the paper goes through all of the panspermia astrobiological, which fits like a glove with Darwinian evidence and with Lamarckian evidence, and convinced me that Chandra was right. We had to write Lamarck and panspermia because, you see, it gives a raison d'etre, that is, a reason to exist for Lamarckian processes here on Earth. You see, let's just put ourselves in the position of a cometary fragment hurtling through space, which has got living material in it, right? But protected from the radiation because it's within a within a protective matrix, hurtling through space at 70,000 kilometres, an hour. I think it's about 20 kilometres a second. But the point is, these fragments, when they hit a congenial niche, let's say something like the Earth or another planet or another water world somewhere, when they hit, there is a Darwinian requirement. So Darwin's right in this sense. They've got to take immediately or die, don't they? They have to adapt to that new environment, whatever the organism are, or die. Now, It's Lamarckian mechanisms of rapid acquired genetic inheritance that allows them to adapt, you see? So it now gives you a raison d'etre for Lamarck on Earth. If the universe is teeming with life, how could it spread throughout the universe like that? And
it has to be able to adapt. It has to be able to adapt.
It has to be able to adapt. So there's a Darwinian selection window for sure. So Darwin is part of the Lamarckian theory now. Do you see what I'm getting at? Okay. It's not the overarching theory. And
I think I do.
And the rapid adaptation is essential now for that to take. If it doesn't take, it doesn't take. If that means that's not a congenial niche. But some of the organisms might. I can give you a graphic just so story that they all invent, by the way, for life on Earth, involving cosmic octopus, if you want to hear that later. But the point is...
I looked at the paper on the cosmic octopus. It's pretty wild.
Well, I'm not just throwing it away. We've published all this. I'm giving the reasons why I'm using the word cosmic octopus. The point is that the embrace of a universe teeming with life allows one to understand what the more correct and more truthful evolutionary mechanism is on Earth, and it's Lamarckian, subsuming some of the principles of Darwin. But it's like in a way Einstein subsuming Newtonian principles. But we're still correct. But now in the Einstein theory of relativity, they were subsumed under a bigger theory. Do you see what I'm trying to describe here?
Yeah. So again,
is a bigger theory than just.
I'm going to summarise this if I can from that lay perspective, and once again, you jump in and correct me where I'm wrong. Okay, so Darwin's theory says that. Under Darwin's theory, let's just say, evolutionary change is incredibly slow because it can only proceed at the rate at which humans reproduce, once every, so you got like 20-year generations, right? And that say an organism develops some, an organism during its lifetime engages in perhaps some adaptive behavior that makes it survive better in some environment, but it can't, anything that it develops after the time it was born cannot be passed on to its offspring, right?
That is the Weissman dictum, yes.
Yeah. And so if some animal figured out a better way of, grazing on some particular grass, I don't know, it's probably a bad example. Some animal a tool, it's basically the ability to utilize resources more effectively. Okay, that's what makes organisms survive better.
Get that knowledge onto your progeny straight away. Get that knowledge onto your children.
Yeah, and so it would not be able to. I mean, you can for organisms that actually nurture their young, they can educate them about what they've learned. But what Darwinism says is you can't actually pass an adaptive trait onto your offspring.
Exactly.
If it's something you picked up during.
Well, look, all of the evidence shows that the Weissman barrier is selective and permeable. Let me give you a graphic example that everyone can relate to. We all think, and Samuel Butcher thought this for certain, that the range of instincts we have, like falling off at sleep, nodding off of a branch, or being frightened of snakes or particular reasons. Why do we have those instincts? Why do we have certain things that we are just instinctually, we don't have to be taught them, we just know that they're in danger? Well, let me give you a graphic experiment that was executed 10 years ago. It was a Pavlovian behavioural science experiment at the molecular level, the odorant receptors, right? The pick up smells in the place. And to really strip it down to basic simplicity, they trained, animals, I think it was mice, they trained them on a certain odour that had a penalty response. It was a very drastic penalty response in terms of the behaviour of the. So they trained.
Smell this, something bad happens. Yeah.
On this odour, with just danger. Don't touch it in terms of food or whatever. And then they bred the males and the females. It didn't matter. It went through the male or the female. They bred them to normal parents, and elements of that instinctual reaction, did that crop up in a progeny? And it did. I'm reducing the experiment now to in a language that people can understand. But we review that evidence in Lamarck and panspermia, like a whole lot of range of other evidence. So the inheritance of acquired instinct, right, in human beings is a classic example of Lamarckian inheritance. Think about it, because it's preparing the progeny for the same danger that the parent confronts.
Exactly. Now, so how the Darwinians have, that's probably an oversimplification, but how the people who sort of, cleave to the standard line have attempted to explain this is that it's epigenetic. Okay, so it's not that.
Sorry, you go ahead.
So because I've seen that as an explanation that's given for, for example, why children who are conceived to mothers who were undergoing starvation or something like that, certain epigenetic changes that pass on to their offspring. This was first observed in the offspring of women who underwent the Dutch hunger winter, where their offspring.
Classic example of that. Yes.
So in other words, they're saying, well, the genes aren't changed, but the expression of the genes is changed. So that's where
the instinct experiment I just described refutes that.
Yeah.
It wasn't just epigenetic, and you're really getting now to the core of a paper that we laid it all out actually in Lamarck and panspermia. We called it. This is Robin Lindley. The original ideas started with Robin Lindley and then refined by me, and then together, and then in our big paper, Lamarck and panspermia, she's a co-author, but there's a whole section in one of the figures there called the epigenetic genetic coupling. The soft version of Lamarckian inheritance is just what you said, the epigenetic, which is thought to be reversible, like within a generation you can reverse that, right? Okay. But clearly phenomena like the transgenerational effect of acquired diabetes, for example, where it doesn't reverse. And unfortunately, amongst our Aboriginal people, it can get reinforced by mating without them even knowing it and making it even worse. And that's when it becomes genetic. So how does it become genetic? So the theory that Robin was groping towards, which I helped refine, the epigenetic coupling theory, we put it out in our paper, Lamarck and panspermia, and we'll probably publish it again now that there's more evidence in the last 5 years. We were talking about epigenetic and Lamarckian inheritance, okay, which is the standard view of it, by the way. And again, it's done to negate everything else I'm going to say because they don't want to know about it. It's the same phenomenon right at that interface. The epigenetic genetic coupling, the basic model which we're fleshing out now, and we outline it, up to a point in Lamarck and panspermia, is that the first phase of a Lamarckian adaptation is in fact reversible. It doesn't appear as if it's going to get to the next generation. It's predominantly epigenetics, and epigenetics, I'll speak more slowly, epigenetics is what are the molecules, the hormone molecules, signaling molecules that bind to promoters and expression sites on genes, which regulate the expression of genes, okay? And methylation and demethylation are really important part of gene silencing and non-silencing, but that's only a small part. There are many other epigenetic processes, and there are new ones being discovered all the time, and they're to do with the regulation of gene expression, right? That's what epigenetics is about. It's not supposed to be inherited. The trouble is, if it goes deeper than that, and it couples with the reverse transcription step, incorporating a somatic, mutational, benign, adaptive change in the cell. Well, that's the key, by sustained simulation, because everyone agrees that sustained simulation can lead to solid acquired inheritance. But how? The epigenetic genetic coupling is into 2 steps. We outline it in our paper, Lamarckian panspermia, clearly. There's the soft Lamarckian reversible phase, and there's the hard Lamarckian inherited phase involving things like, you can't avoid it, reverse transcription. That's why I've been working on it for 50 years. And the cancer cells, by the way, mature, advancing cancer, all have this signature written all over them, by the way. Every cancer cell that's been sequenced, and there's thousands and thousands of genomes now sequenced outside antibody genes, which I worked on, where the original idea came from. They show this, what's called the deaminase-driven reverse transcription signature, which is the one I'm just talking about. And it's to do with mutation at the RNA level, feeding back into DNA by, the reverse transcription copying of RNA to DNA. So the point is, it's epigenetic, all right, but that's the first phase of an adaptation, which can in fact be reversible. But if you keep up the sustained stimulation, and then you have inbreeding, which, for example, goes on in small closed communities, or even not just inbreeding, just small communities where they don't have any choice outside it, like an isolated Aboriginal community. They don't have any choice of a partner outside it, but they're all suffering from acquired induced diabetes or alcohol fetal syndrome. And
we see this in like Ashkenazi Jews have this incredible concentration of genetic diseases. Yeah.
Yes, that's right. And I know it's inflammatory what I'm talking about, but it's the pure structure of these isolated communities. It happens in island communities as well. They get a general exposure, leads to a metabolic malady, and then you reinforce it by people not even knowing they've got it, and then breeding, and then you can lock it in as a genetic malady. This is the tragedy of the Aboriginal people in isolated regions and in many island regions. I've tried to turn the mainstream around on that, but they're not listening to me. They just don't want to know about the evidence. For example, there are clear experiments in rats going back 40 years now, where if you induce diabetes in the beta cells, right, knocking them out, and I've done these experiments, by the way, and seen similar effects, not to the same extent as other people have, but I've seen them. I've done these experiments, but I've seen the better experiments done by other people. And there, it's clear, induced diabetes set up in the male or female line in the initial generation can be transmitted in part or in total to the next generation. And if there's any inbreeding going on, that is, by inbreeding, I don't mean genetic inbreeding, I mean breeding with other people that have the same induced malady, right? Metabolic.
When you say induced, in lab rats and mice,
it's like a chemical that knocks out the beta cells.
Yeah. This is not like acquired through eating crappy food. This is like use streptozone or something to knock out the beta cells.
Yeah, that's one of the ones I used. Yeah, but there's also aloxin. But the point is that these are experimental situations. You've got to understand.
Yes.
But because the phenomenology is easy to understand and see very clearly, it would almost certainly apply to humans. Almost certainly. I can't see it not. Because, rats and mice are vertebrates like human beings are. So, so the Lamarckian theory of evolution, you see, which covers a wide range of phenomena, but there's a lot of evidence for it. And that's one of the points we make and argue in the paper, Lamarckian panspermia, that both the panspermic hypothesis and the Lamarckian hypothesis have gone through real experimental testing, unlike many other speculations, and have survived all those tests. That means, as an objective scientist, again, I come back to the objective, you've got to start accepting it as real. You see, but we haven't even got to that stage yet. That is the problem. Anyway, that is the battle on Earth, on evolution, and in Lamarck and panspermia, we try to resolve it and say, well, this is why these things happen. Because, living systems travelling through space have to adapt rapidly and quickly to their environment. And if not, they die. If they do adapt quickly within one generation, their progeny benefits from that adaptation, which shouldn't happen according to the Weismann barrier. Okay, you see what I'm getting at? I'm putting in simple terms now. So that's why that's really clear.
So really, So I imagine you would have some sympathy for, my readers who, write these comments that are critical of Darwinian evolution. You would actually have some sympathy for their position and say,
I agree with it.
point. There are weaknesses. There are big, fat, gaping holes in this hypothesis. But there is work that has, if you like, filled those holes. It's plugged those gaps. And it's not that you're rejecting Darwinian evolution. You're saying it's not the full story. There's more to the picture. There's more to the picture. Yep.
It's more detailed now. It's detailed now. You see, back in the 70s, when poor old Fred and Chandra, they actually believed that Lamarck was dead in the water themselves because everyone was attacking him. But that's when I started my career. I'm not trying to draw any accolade. I just know I started right in the ground floor of that. And therefore, and Reg did too. Reg is still alive. He's at the University of Toronto. And we've stuck it. I stuck at it probably more than Reg, but Reg did critical experiments on just the sort of things, the Pavlovian conditioning and then breeding. He did a lot of critical experiments like that. I've given you one on the odorant receptors and the development of instinct. Well, Reg has worked on that experimentally. So the point is, to sum up, the Lamarckian theory now makes far, and based on evidence from Earth, makes far more sense in a panspermic universe than it would in a Big Bang. Life started on Earth only started on Earth and then propped up suddenly all over the universe. It just makes much more sense. It picks off Occam's razor's point of minimising the number of assumptions you have to make in an explanation.
You bet. So, okay, tell me when you want me to put up the rainbow worm slide.
I think we're ready to put it up. Why don't we put it up? And
let's do the rainbow worm. Okay. What's the rainbow worm? My goodness.
Well, I'll get to that in a minute, but let me just talk about the general things behind this, because let me talk about, and we discussed this. We don't discuss the rainbow worm in Lamarck and panspermia. This is too hot to handle. This is posted. I've posted some of these concepts, not the full deal, at some of my posting sites, but that's not published. No standard journal now would accept anything like this. But let me just go to. I was going to say something prior to that. Anyway, the point is Let me go to this, and it will trigger it. This slide was presented for the first time 5 years ago at a private symposium that we held for Chandra Wickramasinghe in Melbourne, when I was living in Melbourne, at the Blackman Hotel on St Kilda Road on Sunday the 8th of December 2019, just before we became aware of COVID. We're celebrating his 80th birthday, and it was a private symposium of people who, his brother was there, Dale was there, I was there, Dr. Robin Lindley was there, a few other people that came in by Zoom, were celebrating, and Genzuko Tokoru from Japan, who helped fund it. We were celebrating his birthday and the fact that the universe was teeming with life. Now, a week before, I was one of the organisers for the meeting. And the week before, my sister died and passed away, and I went to her funeral, and I was much struck by how much, compared with her brother Ted, she was much loved by her community. But she had many, many people there testifying what a wonderful person was. You know, her religion, by the way, was most unusual. That was one of the reasons why I suppose she was most loved. She was a genuine Hare Krishna, but very spiritual. But what I noticed about that meeting, that funeral, was that in the room. But the other thing that it took me back to my mother's death, 40 odd years ago, and 35 years ago, and how cold she was. She was a vital woman who went down with Alzheimer's, like many do. A vital woman in her life. She inspired me. But in death, when I kissed her goodbye, it was cold, just cold, and there was just nothing emanating from her body. And it had a big impact. That event had a big impact on me. I think medical people see it all the time, but it was my first time I'd seen a dead body. Okay, it was my mother.
Yeah, you brought to mind a really vivid memory that I have, exactly the same experience when my father died, and most of my family members didn't actually want to go to the viewing, but I did, and yeah, it's so striking, isn't it? This person that you've known and loved, and you touch them, and they're cold. Obviously, they've been in.
It seems to have gone completely.
Yeah.
Well,
yeah.
Look. I'll get to that in a minute. We'll keep the slide up. Let me just I've now got what I wanted to say before. In Lamarck and panspermia, I keep coming to the paper because to me it is probably the most important paper I'm ever going to write. Okay, I just know it is. I can't excel. Some other data will come along and add to it, but in terms of bringing together the concept, in the middle of the paper, we not only talk about demarcation evidence and meteorites and all that, but we talk about the boundaries to our knowledge, our scientific knowledge. And when it comes to the origin of life in the universe, we have real boundaries, and many scientists have not confronted them. For me, if you confront them, you recognise them as boundaries, and there's simply no point in going beyond them, except what I'm going to talk about in a minute. The first boundary is the upper boundary of the size of the universe. Now, we didn't know it back then, but it was already pointing in that direction. In 2019, we know that the universe now is certainly not a Big Bang universe. The James Webb telescope has shocked the physics and astronomy community, completely shocked them. They viewed out beyond the 13.5 billion light year boundary, and what did they see? They saw more galaxies. As far as as far as the eye could see, more galaxies. That shouldn't have happened. That prediction refuted the Big Bang theory as the standard theory as a. But do you think they're accepting it? No, they're trying to explain it away. It's a standard, traditional dogmatic response from scientists, by the way. Anyway, for me, that kills off the Big Bang theory. So we're probably dealing with what Fred Hoyle, Herman Bondi, and Tom Gold predicted back in 1948, that the universe is in fact an infinite. The steady state theory of the universe. That's the first. And
infinite, of course, infinite is beyond human comprehension.
It's eternal. It's always been there, and it will never end. Now we can't. A logical scientist in the human training like myself cannot grasp it. I cannot grasp it.
I'll tell you what my husband said. When we were discussing this. Well, I was talking to him about my upcoming interview with him and these competing ideas of life on. Did it develop here? Did it come from from somewhere else? Whatever. And he just said to me, I don't even know why people bother asking these questions because you can't answer them.
And we have a very similar attitude, but as a scientist, I'm forced to reach that boundary. But that's the upper boundary. The lower boundary is even more interesting, and it's been around for over 100 years. It's the quantum world, quantum weirdness. Again, I published another little note that'll. Sorry, this is it. No, another little note that'll hasn't appeared in a journal called "Quantum Weirdness and Living Systems." But when we get to the quantum world, below the level of the atom, or at the atom and lower, everything's different. The world is so weird. All the rules of logic, cause and effect logic, everything has changed. Action at a distance, it's all gone. In fact, modern quantum computers, they want to be developed because they will be able to work. Once we work out how to run a quantum computer at normal room temperature, it will change the world. It will. As I went to a conference 20 years ago where a CIA specialist was freaking out over. He said, "When we a fractured American accent, when we invent quantum computers, nothing will be private. We will be able to penetrate every internet, email, everything at once, and it'll be all over. There'll be no such thing as national security." In a sense, he's probably right. But the point is. The quantum world, we don't understand the logic at all. Now, Richard Feynman, a famous physicist who got the Nobel Prize for quantum, chromodynamics. He said to everybody, he tried to bring everyone down to earth. He said, look, anyone who thinks they understand quantum mechanics is crazy. No one understands. And this is the guy that added to the whole phenomenon. What we have are operational rules, statistical rules at the quantum level, which seem to work. The Schrödinger equation. My son Julian's a quantum materials physicist, and he says, developing a theory from the elements, which they do, theories, but from non-living matter now, is a standard part of quantum mechanics. But we don't understand it. I defy any physicist to explain it to me in a language that a human mind can understand. We can't understand it. So there are two boundaries to our knowledge, as far as I'm a bit like your husband. There's the lower world, the quantum world, and there's the infinite world. There are two boundaries which we'll never, ever be able to understand. So I've reached that point. I said, but I've also reached this point with things like COVID, which was clearly a disease from space event. The near-Earth neighbourhood and looking for life in the near-Earth is a very legitimate, practical, pragmatic thing to do. We can do that. But trying to get these bigger questions and answer to them, I don't think we can. Now, I might have a dispute with people, but look, you're now coming to the point where every individual scientist and person has to make. Everyone has to make this decision. And this is the thing that struck me after my sister's death and the memory of my mother's death. Talking about panspermia at a panspermia conference. This was my last slide, and I'm actually reading Henri Bergson's 1907 book right now. I should have read it years ago. It's very tough going, that. I wouldn't. Anyone who's interested should try reading Henri Bergson, but it's tough going. He's a philosopher and a mathematician, a Frenchman who lives on a totally another plane. Okay, he really does. But he came up with one of his essential principles of life called the Elaine Vital, the life force. Now, after my sister's death and my memory of my mother's death, and knowing what I know about the universe and those boundaries I've just said, I started to think like a scientist about various critical phenomena about life and death, difference between inert matter and living matter. Now, this woman, Dr. May Wan Ho, whom I knew a lot about, who was a strong supporter, I could see why, of our work on Lamarck. I knew her in a most productive phase of her life, but she produced a remarkable book called The Rainbow and the Worm, The Physics of Organisms. It was published in the 1980s. It went through many reprints and impressions, and it is in fact a big scientific bestseller. It's very rare for books rich in data, like Fred's book, Cosmos. It's very rare for those sort of books to have a big mass audience. But The Rainbow and the Worm does have a mass audience, and it became a bestseller, The Physics of Organisms. But the most penetrating thing in the whole book, for me, amongst many interesting issues that she discussed on, the physics of organisms, was this one. And I'll describe, it's an experiment, so I'll describe it now in the context of my knowledge other stuff that I've just meant. So that's the title. In that little photo at the top, and in many photos other photos like that she had in the book, that is a Drosophila embryo. It's a small microscopic. Right? Yeah, Drosophila flies, right? That's what people work in the lab, standard flies. The trouble is, all embryos have these. An embryo from one organism is often very difficult to differentiate from another. They look the same, right? They look like little worms.
Oh, totally. Right? Like tadpoles, right? Until we start to develop into something recognized human.
Well, that's metamorphosis extraordinary, isn't it? You think about it. When you think about metamorphosis, but anyway, let's just deal with. They developed techniques to pick up biophotons. That is, these are photons of light emitted by living systems. Living cells produce their own light. Okay, there are a lot of implications for that, by the way. But that's one of the things they do. Now, if you look at, let's just read it through. So this embryo, the Drosophila embryo, is viewed under a polarizing microscope. And this is the key discovery they made. It would have been garden variety for everyone else, but for me, it just hit me between the eyes. I couldn't believe it. She said, we discovered an imaging technique that enables us to see all the colours of the rainbow in a living, crawling, first instar stage Drosophila larva. So she described, right, that was the first thing. They had all this battery of knowledge. Now, biophotons, by the way, those colours, other people said it's a fact of biophysics. All living systems, it's probably the cause of the auras that we have around ourselves. All living systems, human beings, all
I was going to say, this is so reminiscent of the concept of the aura. Yeah.
Yeah. Okay, well, that people are aware of that, right?
Yeah.
Now, on the next thing that they discovered was that on cryopreservation, the colours fade, then recover when the orbitin revives. By that I mean when you freeze it down, below -40 and then thaw it out slowly, the colours come back, right? That's another remarkable thing about life. Why can you freeze and thaw them? See, that's another example of panspermic survival in the universe. How can you freeze and thaw them?
Indeed, yes.
And in vitro transfer and embryo transfer, we do it all the time. We freeze eggs. We freeze -80. Then we thaw them out, and then we put them into a
living uterus, and then you get a baby out of it.
So can tolerate this wide range of incredible insults, if you like, physically. But the point is, on cryopreservation, the colours dim right down. They don't go away. They fade, but then they completely come back and full blossom when I thaw them. Okay. But the third one's the killer. Sorry, I shouldn't use this like that. The third one is the crunch scientific explanation. On death, the colours permanently fade. Basically, they go pretty fast. And the smaller the entity, the less complex, the faster they go. Because we've got a really complex cell. You've got many parts of it that die more slowly, and your hearts keep beating, other things die more slowly, complex. But in a smaller organism, it looks like it's happened straight away. Now, that I've been thinking about that result for many years. And knowing what I know about the way biologists, chemists do things in the lab, because I worked in a lab like everybody, everyone would agree that if you could use, say, a mass spectrometer or some other technique to figure out the exact chemical composition, that is, count the number of atoms, all the components of the larva while it was still alive, just get a quantitative number. It might take a big computer to do that, but if you, in theory, it's theory because this is the way biologists think about living matter. It consists of an assembly of atoms and molecules, right? If you could measure them exactly, quantify them, then after death, the moment after death, when all the biophotons go away, you'd find the same equivalence. That is, the same atoms would be identical. They'd be identical. So where did this force that gave rise to the biophotons, where does it come from? Well, the most immediate one that comes to mind is that in living cells, complex living cells, all the electrons get shared over other molecules. There's electrical currents shared here and there. So it's organised. Okay, a healthy living cell, healthy embryo, the electrical properties of matter are highly channeled and organised. Think of all the different processes. You've not only got DNA replication, RNA synthesis, protein synthesis, but metabolic control. So many different processes are all bound to be controlled. So it's organised. So I then, after my sister's death, I asked this question: where does the organised electrical energy, for example, emission of biophotons, variable wavelengths, go on the death of living organisms? Pretty garden variety question, actually. I want to know where they go. And then I said, what would a standard mainstream physicist think about this? Well, the standard mainstream physicist believes, quite correctly, in the conservation of mass and energy brought together in Einstein's E = mc squared. Okay, but where do the biophotons go? So I immediately made the leap. I've tended to do that a bit in my. I thought, well, this might be Henri Bergson's Elan Vital, the vital force of life, the organised electricity of human beings, of living systems. So I then, well, if a physicist can create an equation like E = mc squared, I can put equals F. life force. And then I can put another equal sign and say the soul or the spirit. That's why I'm ending on that point.
So where did your sister go when she died and her body was left?
Well, her spirit was in that room. I could feel it in that room amongst those deep.
Yes, but it was not inhabiting her body.
Yeah. Where did it go? Where did it go from my mother's body?
Yeah.
Where did my mother's personality go?
Yeah, I can tell you too, as someone who has gone through naturopathic training and so has been exposed to a broad range of, let's just say, traditional notions of human health. The concept of qi is absolutely intrinsic to traditional Chinese medicine. In Ayurveda, it's prana, and then the physiomedicalists, the sort of 19th century naturopaths and herbalists called it the life force, and then Hahnemann in homeopathy calls it the vital force. So there have been all of these systems of healing that have acknowledged that there is some principle, there is some animating principle that organizes, if you like, all of these random atoms into a living being. And what you aim to work on or to work with, if you're a practitioner of traditional systems of healing, is you're working with that life force. But of course, in medicine, there's no acknowledgement of that life force. I mean, clearly they recognise death, right? Well, once he's dead, you can't work with him. But there's no attempt within medicine to actually acknowledgement of life.
Sadly,
go ahead.
But then again, for very practical reasons, they can't be occupying themselves with the afterlife all the time because they're trying to keep people alive. So, it's very practical. There are a lot of reasons for it, but I think we're raising them now, these key issues here. Anyway, so the soul is thought of as eternal. So where do the souls go? Where are they located? Well, they're certainly not in this world. They're probably not even in this universe. I'm just speculating like all human beings would when confronted with this stuff.
Absolutely. I figure if you've never asked these questions, I don't know, maybe you need to start. But this, obviously there are enormous challenges to, let's just say, established religion in the idea of panspermia. And of course, really hardcore creationists are they strongly object to the idea of evolution, because in their model of Earth and God and humans, God created Earth, and Earth is a special place, and humans are special because only we have this sort of magic.
Stephen Meyer, you probably know The Signature in the Cell. You may be aware of it. 10 years ago, published this famous book, The Signature in the Cell, and it's basically going, just talking about all the things you've just mentioned, how it doesn't make, and all the issues I've just raised. I agree with Stephen Meyer up to a point, but I disagree with him because he doesn't want to mention panspermia. So in the whole book, they don't want to mention oil and Wickramasinghe. And well, that's strange. He puts himself up as a scientist, but he won't talk about one of the other major scientific developments. So it's emotional now. And when I say from the left and the right, they smear you from the left and the right.
Yeah. So speaking of major scientific developments, one of the things that I was absolutely fascinated to encounter as I was, reading around this topic to prepare for speaking to you today is that Francis Crick, as in, one half of the famous Watson and Crick, the men who probably actually stole a woman's idea in coming up with the idea of anyway, so Francis and Crick are famous for developing the idea of this double helix structure of DNA from which all studies of genetics have proceeded. In other words, this is a pretty big deal, and once again, these are serious scientists. And so I was fascinated to find Crick, a paper by Crick, France, again, one half of Crick and Watson. So, but he co-wrote it with Orgel, who I'd never heard of, and this is the directed panspermia hypothesis. So, hang on, let me just pull this paper up. So the directed panspermia hypothesis is that directed panspermia, the theory that organisms were deliberately transmitted to the Earth by intelligent beings on another planet.
That's possible. I'm not going to deny that. I'll tell you, can I give you why Crick and Leslie Orgel? Leslie Orgel was a famous scientist in his own right, and they teamed up at the Salk Institute when Crick shifted to California. See, he was the general molecular biologist that all the developments in the 1960s and the discovery of the genetic code, and he was the Fred Hoyle, if you like, of molecular biology. He directed developments, and with Watson, who did amazing contributions as well. Look, they were really big, important scientists, and all my training depends on what they did. But he reflected on the universality of the genetic code. Now, for those who aren't biologists, the universality is the correspondence between the DNA or RNA sequence and the protein sequence, right? They're two different molecular structures. But the code is triplet. There are 4 nucleotides or 4 bases, A, G, C, and T, and uracil replaces T in RNA, right? But they're four in principle. And then they're organised to code for the 20 proteins, or the 20-odd amino acids. They're organised in triplets, right? It's a triplet code. And it took about... So
you've got three of these nucleotides that code for one amino acid. And I think there are some amino acids where there are actually a number of different base pairs, a number of different triplets.
Yeah,
right?
yeah. Well, I was going to try and go to my computer to show you what the genetic code looks like, but it's like, so the triplet in the DNA or RNA and the amino acid it codes, one of the 20 it codes for, it's very redundant. There are 64 possibilities. So given there's only 20 amino acids, it must be redundant. The same, a derivative of the same triplet can probably code the same amino acid. But the point is, it's universal. All living systems have it. It's a reason why we can do biotechnology from E. coli cells through to higher animal cells. And he reflected on the fact that it had to be universal throughout the universe, which is a reasonable deduction. If it had to be universal, he certainly didn't buy a lot of the standard theories that life began on Earth through ponderously slow Darwinian. He didn't buy that. So Orgel and Crick, came to the obviously logical one that if there was organised life out there in the intelligent life, they could have seeded it here on Earth. That's reasonable theory. Thomas Gold, one of the co-authors of the steady state, theory of the universe, he flippantly said in the early 1960s, 10 years before Crick and Orgel, he said, well, yes, maybe some visitors from another planet here landed here about 3.5 billion years ago and tossed out a few bugs and scattered them around. He actually said that at a scientific conference. So, look, the idea is plausible, but we don't know whether it's true. But knowing what I know now about the universe teeming with life, it's plausible, but you don't even have to go down that route with direct panspermia. There's another bigger picture involved than just directed panspermia. I don't know how much time we've got, but I could talk about cosmic octopus because I'll do it because every other evolutionist has a just so story, just so. Well, I've got one on the cosmic octopus. Anyway, look, I don't know whether you want to hear it. Maybe it's.
No. I think having raised the cosmic octopus, we have to discuss this because this is the corner.
One because we're almost 2 hours now. Okay, so let me just say this, that the prediction of the universe teeming with life is as many other water worlds out. Just in our solar system, Enceladus, one of the moons of either Saturn and Jupiter, I forget which planet, and Europa. They're water worlds. They are big moons, not as big as the Earth, but big, and they are covered with an icy shell, right? Some meters of ice covers them, which harbour everyone agrees there has to be oceans in Encelada and some of the other ones, these water worlds. So they agree that they have. So the external would be subject to, absolute zero. Everything's frozen. But the remarkable thing about these Encelada is they have geysers geysing out, ejecting huge sprays of fluid, clearly fluid, into space. Much like a comet that's incubating life, having their big sprays, and we see it all the time in celestial objects that are incubating something, right? But why are Encelada and Europa doing it? Okay, this is my just so story. Okay, this is my just so story. In Encelada and Europa, there are water worlds, massive oceans, like our Pacifica, of life, all sorts of organisms thriving. Bacteria, higher cells, octopus. And of course, how can they operate in the dark? Well, they can operate in the dark because of these biophotons. Living systems actually emit their own signals, right? And other living systems can see them, with their... But the octopus is extraordinary. The octopus appeared suddenly on Earth 275 million years ago. We don't have any fossil evidence for the octopus, but we have a lot of what's called coalescing phylogenetic evidence for all their different proteins. Where did they first emerge amongst all the panorama of other organisms? And everyone agrees, this is an agreed number. 275 million years ago, octopus and squid suddenly appeared on Earth. Now, then they say, how did that happen? Because the precursors, the nautiloids, very simplified versions of squid, but shells with shells, hard shells, they appeared during the Cambrian explosion, 250 million years prior to that, okay, prior to the 275 million. How could the nautilus, evolve into an octopus? Because the octopus really is a really strange beast. They are highly intelligent animals.
Unbelievably intelligent, real, like fast learning. Yeah.
Yeah, you've got to... I recommend a great documentary put out in 2020. It's on Netflix. It's called My Octopus Teacher. It's by a South African documentary filmmaker. He won prizes. It's award-winning. He's very famous. My Octopus. Just watch that in the context what I'm going to tell you now. Anyway, it occurred, and I didn't see that documentary till after I'd already come to these conclusions. But this is what I thought. Just imagine we've got oceans, deep oceans in Encelada or Europa, thriving with life. We've got octopuses, and their life cycle is short, probably 2 years. But what they do, each female produces thousands, if not millions, of fertilised eggs, right? Literally. As they get old and disintegrate, just watch the disintegration that took place in that documentary with My Octopus Teacher. And they recently saw these fertilised embryos. Okay, that's key. Fertilised. They're not unfertilized, they're fertilised. So in the appropriate environment, they're ready to go.
Yep.
Now,
right. So embryos are in some kind of state of suspended animation, but when they get in the right environment, it's on.
Exactly.
Okay.
So story, and I've called it cosmic octopus and space icebergs. Just imagine in the certain stage of the evolution of our solar system, a massive comet or massive asteroid collided with Encelada, caused a massive ejection of oceans into space, and it would be snap frozen. It's absolute zero in space. Now, so you've got your iceberg with snap frozen octopus embryos, millions of them.
Yeah.
You see where I'm heading now.
I do.
This object somehow or other gets into an orbitary trajectory as an iceberg now, as a frozen iceberg in space, that lines up directly with Earth. It'll hit Earth about 275 million years ago, and there would have been a lot of extinguishing of living forms in it. But with a big object coming in at 70,000 kilometres an hour, within minutes it's in the oceans, right? One of the oceans on the Earth. It's melting and thawing out, and we've got all these fertilised octopuses. Boom! And that's
oh my God.
I'm telling you, we could survive in this environment. Darwinian, all of that doesn't matter. We could survive in this environment. So they start producing more octopuses. And then
yes. Yeah. Okay. So one of the articles that I read about the cosmic octopus, I mean, it was really funny. It was saying, look, this is wild. This is really out there, but hey, there might be something to it. And the final line of it was, better watch out. Our octopus overlords might be contacting us from space saying, hey, we want our eggs back. Anyway, but it seems to me that this does.
Sorry, I'm interrupting.
Yeah.
Keep going. Sorry.
No, that's okay. It seems to me that this does answer again one of the criticisms of the critics of evolutionary theory is the gaps in the fossil record. Like, how do you account for like you've got this blob over here. It's just a blobby thing, and it does blobby things, and then you've got this sophisticated life form. And where's the steps in between?
Yeah.
How does the blob get to the sophisticated?
It's puzzle biologists, Stephen Jay Gould, when he was alive, and Niles Edgeworth, when he arrived, they published a famous theory before Crick and Ogle, and before Chandra and Fred really started hitting their straps. They're all fitted with what Fred and Chandra discovered. It was called punctuated equilibrium. If you look at the entire fossil record and the emergence of life on Earth, there are all these huge gaps. Suddenly a new organism appears. Where did it come from? So, but a traditional evolutionist said, it must have come from somewhere. That's the first thing, the big assumption. So some of its sequences must be related to this. So what they do is they construct a tree. It's total fantasy that's rooted in some origin. I have to be emphatically derogatory now of my phylogenetic colleague. It's total fantasy. It's all made up. It doesn't exist, okay? Because what you have is the correct model, and we discussed this in our first paper of these two big series in Progress of Biophysics of Molecular Biology, and that was the origin of the Cambrian explosion. And we talk about the octopus there at great length. We talk about it again in the second paper, Lamarck and Pounce, but the first paper we go through my description of this space iceberg, and the way we actually published that whole paragraph as a possibility, right? We have got to raise it. So the staggered nature of the gaps in the fossil record have got to be addressed. They're real, punctuated equilibrium now is a descriptive term for the phenomenon of life on Earth, particularly since the Cambrian explosion 500 million years ago. Doesn't really handle much prior to that, but because that itself is one of biology's biggest mysteries. How could cell-based, plant-based, complex life suddenly just explode into existence suddenly? And it's one of the criticisms that a deeply religious person would attack standard evolutionists. So I agree with them. You see, I asked that question: how could that happen? Well, in our paper, the origin of the Cambrian explosion, we go through, is it terrestrial or cosmic? So you can imagine some of the critical evidence we evaluated before, So I think it was a planetoid that struck Earth. Well, a planetoid was a mini-sized Earth-like object that came from deep space, collided with Earth, and just seeded all these wonderful life forms already formed. That's why suddenly it exploded. See what I'm getting at? And it's a real phenomenon.
I totally do. It is absolutely mind blowing. And of course, then we get to the whole issue of panspermia kicking the can down the road. Okay, so scientists and people like me who don't call themselves scientists but are just fascinated by deep questions like this. One of the things we want to know is, what is the origin of life? And so evolution, or Darwinian evolution, to be really strict about it, posits this sort of, life in the warm pond, and then various forces acting on the building blocks of life, and then poof, life, right? And then the creationists say, well, it was God, and God made everything, and like, so what was there before that? Okay, we can't really talk about that, and so. Panspermia doesn't really answer the question of the origin of life. It just says, well, it's out there.
No. And if they're honest, if other scientists are honest, they would concede that. like I've tried to be. I'm not putting myself up as being an honest scientist. Just sort of, if you follow the evidence and the objectivity of it, you have to reach some of the sort of conclusions I've reached. Anyway, it's quick kicking the can down the road, no question, which means it's legitimate for religious explanations to be in the same forum as scientific explanations when it comes to the ultimate issues of the origin of life in the universe. Yep. And I have no answer for it. But you can't negate deeply religious people for thinking that way, because all the evidence seems to confirm, and it does in a way, it does confirm what they've been thinking. And so I don't know what the answer is. It's not going to console me, is it? I actually envy deeply religious people that are certain they know the answer.
Me too. Wouldn't it be nice? Wouldn't it be nice to have that degree of comfort and to not be plagued with these questions?
But it would be, for me, it would have to be highly refined, things. So it's all the things I'm talking about are all my highly refined ways of viewing it. Where does the soul go? Is there another existence? Is there another repository in existence that's conserved with matter energy that looks after the soul?
Because the thing that strikes me, okay, is that and this is where I think the transhumanists are just so off base. I mean, to be human is to be embodied, right? So our experience of life on Earth is mediated through a body. It's this corporeal substance, and so to me, it just given the fact that we are embodied beings, what it seems to me is that it's simply not possible for us in our embodied form to understand these questions, right? To even imagine what it would be like to be a soul, a spirit, or like wherever the biophotons go. Like, we can't imagine that because definitionally we are embodied. That's where I come down on this. Like, I'm not going to know till I die.
Look, it's a massive conundrum. Look, the great philosopher Karl Popper, who guided me and many, by the way, in my early years, he summed it up too, that the world of the quantum and the world of these boundaries are a real conundrum. The original is a real conundrum. It doesn't fit with any of the ways that you would normally have to want to think about the origin of the genetic code and DNA, RNA, protein. All those standard rules. Because one depends individually on the other. They'd have to come into existence altogether. You see what I'm getting at? So he admits that. See, the ones that are admitting it, but then again, I suppose if you have a deeply religious upbringing, it's easier, to handle it. I didn't. I had parents who were. That's another story. But I went to religious instruction when I was a child growing up in Darwin. So I was one of the kids that were on the outer. I sometimes wish that I'd had it because I would have had more knowledge of mainstream religion. But that's the way it is. You can't change the past. But the thing is that it was my father. I'm going to give you a little hint now why I have some way to think why I do. When I was about 4 years old, he was a surveyor. He was also an amateur astronomer. He knew a lot about the universe. He knew a lot about Fred Hoyle, Herman Biondi, and Tom Gold's theory of the steady state. He knew it. I only reflect now on the things he told me. He'd take me outside and to all the various constellations in the sky. He'd have his own.
My dad did the same thing. We would go out in the backyard, my brother and sister and I, and my dad had a star chart. Just at the time of year, and we'd look up through Dad's binoculars, and
so he was that. At a very young child, he was talking about an infinite universe to me, and I made the deduction. I don't know, sometime before I was 10, there must be another Ted Steele out there somewhere.
That's
In infinite universe, there's everything is possible.
So this is the multiverse idea, that there are just multiple. Yeah, okay, that's.
But look. Anyway, that's I've given you. It was in my life in some ways because I pondered it. I used to come up with, experiments. As a teenager, I know how we could refute that crazy idea of mine. Refute God. We could make life in the test tube. Of course, we can't.
Yeah, we can't. We can't.
Yeah. But look, anyway, look. So all of this work, all this research work after 55 years has led me to this position. It's amazing.
Yeah, that's the most extraordinary journey. So Ted, we've been talking for over 2 hours, and I mean, this has been one of the most fascinating conversations of my life, which I knew it would be just from reading your papers and the work that preceded yours. And so, how do I even summarize this? Okay, so for those people who have looked at mainstream evolutionary theory and said, there's some pretty big gaps in this, a lot of this doesn't make sense, you would say, you're right, I agree. There's some gaps in mainstream evolutionary theory big enough to drive a Mack truck through. You would also say that in the absence of any better explanation, the presence of God is as good as any for the fact that we have life. Life is not unique to planet Earth, and not just life as we know it, Jim, but life in a form that we would find recognizable is everywhere. It's everywhere in the cosmos. The cosmos is in fact teeming with life, and we can explain these kind of gaps in the fossil record and many other issues that very, very intelligent, sensible people have identified within mainstream evolutionary theory. We can actually fill in those gaps with panspermia and Lamarckian inheritance, and in fact, those kind of
makes sense. Yeah, make sense of it all. Yes.
Yeah, but at the end of the day, we puny embodied humans cannot hope to really understand life or its origins.
I'm afraid I haven't given up. I just know that it's not possible. It's not possible for us to understand it. As a scientist, as a practicing scientist, that's built my whole career on objective observer and independent evidence, that this is where it leads to. So I have to concede that you can't. But I wouldn't be an extreme creationist as well, who lambasts the other. I would say there's a lot that's totally credible in what they say, and really looking at all the evidence. It's just that I just hope they wouldn't delete some of the great scientists like Fred Hoyle and
I don't know. It's like God made scientists too, so there's room for that. Okay. All right, Ted. Thank you so much for being really generous with your time. I'm going to have you send me links to papers that you mentioned today, and maybe names of books that you've mentioned, because I think there's going to be at least some people who are prompted to look more deeply into this as a result of our conversation. And it would be great if you could provide kind of like a starting reading list. So just send me that afterward, and I'll put it in the show notes for today.
Yeah, right. When do you want me to do that?
Whenever you've got time. Yeah, it'll take me a while to edit this. So
okay, yeah. I'll make a note that I'll send the list. And I'll keep it short, up to about 6. You don't have to read endlessly.
Perfect. That'd be great. And are there any other thoughts or questions, or I don't know, anything that you'd like to leave our listeners with today on this subject?
No, I think you've taken everything out of this sponge. I feel like I'm completely depleted now. I've never experienced an interview like this before. The snippets and bits, but the coherence of the interview has given my complete position out. No doubt you have to edit the dogs out and all that sort of stuff, but that's all I want to say. Thank you. I haven't ever had a chance to do this in a more relaxed environment.
Well, this is so gratifying for me. It's real, and I find that, number one, extraordinary that you haven't had anybody else, who wanted to just sort of draw this information out. But I guess this reflects back on our earlier topic of conversation, which is that the incredible rigidity of thinking of people within science, and the science communicators, like the hangers-on, who are kind of like the court historians. These science communicators are the science world's equivalent of court historians, who sort of tell the mainstream version like that's their job. Yeah.
Let me just qualify one thing. I have met someone during COVID, and he's a prominent businessman here in Queensland. I don't want to necessarily break his cover, but he accepted, because his trade with China depended on him knowing what the hell was going on in early 2020. He accepted completely the explanation for COVID, and that allowed his business to survive, because he took countermeasures completely differently from the madness that enveloped during COVID. He did the complete opposite of what he was being told to do for his business. It was quite a big business. It wasn't small. And so, yes, I do. And I don't necessarily want to blow his cover now, but because I haven't asked his permission. But there was one other person who instantly grabbed the idea immediately. And I didn't experience that also when I was actually talking to other people during COVID. So this is outside my scientific circle who do accept it. Like, I'm talking about someone completely outside, from a different walk of life, accepted it completely because it made so much more sense than the other ones. Anyway, I just want to qualify.
And isn't that the purpose of science, to help us make sense of the phenomena that we experience every day. All right. Once again, Ted, I want to thank you from the bottom of my heart. This has genuinely been fascinating, and I after I've digested some more material on this, I might well want to get in touch with you again. And actually, too, if my listeners have any questions, it would be great if you could come back on again. But we'll tee that up subsequently. So yeah.
You've got everything
Wrung it dry. Awesome. All right, Ted. Thanks once again, and I will definitely be staying in touch because this is just endlessly fascinating.